So we finally get away from talking about chemistry and get to actually start talking a little bit more about things that are directly biological when we introduce Chapter 4, which is the chapter about the cell. Cells are obviously the smallest and simplest unit of life. You've seen that in the previous lectures, and again I'm noting it today. So that's one of those reminders to you that these are things that you may want to be aware of for your exam. Again, I get very excited about the fact that there's some wonderful historical perspective here. We, throughout most of human history, never knew that cells existed. It wasn't until microscopes were actually developed, initially in the 17th century, and then really later on in the late 17th century, and much later into the 19th century, that we were actually able to see microscopes. So Galileo was the person who was credited with inventing the first microscope. A gentleman by the name of Robert Hooke used a microscope to observe cork and other things, and he termed the coin cell when he looked at cork because he saw the empty dead cell casings from cork, and it looked like the cells that monks lived in, and that's where the term cell actually came from. So it actually was more related to a religious term than it was initially to a biological term. There was a Dutch dry goods merchant named Anton van Leeuwenhoek who was the first to see living cells. He wasn't a member of any of the important scientific societies, and so he would send his drawings of cells that he saw by setting up a system with well-ground lenses to the scientific societies. No one believed him for quite a while, but then were astonished with the first living things that he saw. He was also the first person to see living things moving. Robert Brown in 1820 was the first to identify a nucleus, and it wasn't until 1850 that we came up with both Schleiden and Schwann and Virchow leading to the formulation of cell theory. Now obviously cell theory is one of those things that you'll have to know. You'll note that there's asterisks on this slide here. It has three basic premises. That all living things are composed of cells. The cell is the simplest level of biological complexity, the smallest living things, and the basic unit of life. We also say that all cells arise from pre-existing cells. The dilemma we run into is, where did the first cell come from? And again, we have some hypotheses about where that is, but again, we only have hypotheses. There's no way that we'll ever prove where the very first cell came from. In order to see cells, again, it was very important to have microscopes, so it's worth taking a moment to talk about microscopes. The microscope you used during the first week of class in laboratory was a compound light microscope. You note that everything that we looked at had to be squished between a slide and a cover slip, or thin sectioned, and so it does limit what we can see. There's no three-dimensionality to what we see, and it can only magnify up to 1,000 times, which seems like a lot, but isn't enough to see all the structures. A transmission electron microscope is the microscope most likely to be used by biologists to see high level of structures. So if you wonder why the pictures of cells that you see in labs are different from what you've seen, is because you weren't able to use a transmission electron microscope, and that's where some of the fine detail that we have about cell structure comes from. And finally, a scanning electron microscope gives you 100,000 times magnification, again using electrons but bouncing them off the surfaces of images. Gives a three-dimensional view. So here is some scanning electron micrographs of structures from a Shasta daisy, and these were taken by Kristen Lennon, who is one of our staff members. But these are the reproductive structures of the Shasta daisy. And again, transmission electron microscope images that she took of the same structures. These were thin-sectioned, very thin sections. This is something called a pollen tube that the pollen uses to transmit or to make its way down for fertilization. These are two pollen sperm. Some little structures you can see here. This is a mitochondria. This is a Golgi. Also some vesicles. These are things you can only see when you magnify. Literally, you have to magnify more than 100,000 times. Okay, so we're going to go back to some of the things that we already discussed, prokaryotic and eukaryotic cell structure. Again, this is a reminder that we just covered it briefly in the first chapter, and now you really just need to know this. So prokaryotic cells were the first to evolve. They were the most primitive. They don't have a nucleus, but they do have DNA that's located in a nucleoid region. There are no membrane-bound structures or no structures that are enclosed with a membrane that carry out particular functions, but they do carry out many of the same functions as eukaryotic cells, just not necessarily in specific areas. There is a limit because of their size and because of their lack of organelles of what any prokaryotic cell can do. Eukaryotic cells, we believe, came to rise long after the prokaryotic cells. They're more complex, they have a true nucleus, and they also have membrane-bound internal structures called organelles. So here's a nice picture of a prokaryotic cell. Again, when you look at it, you don't see any particular membrane-bound organelles, although there is a nucleoid structure. You can see there's a plasma membrane and a cell wall that surrounds this. This cell also happens to have a capsule. The picture has flagella, and you can see these little dot-like structures are ribosomes. Those are non-membrane-bound organelles that are found in both prokaryotic and eukaryotic cells, but they're a little bit different from prokaryotic to eukaryotic cells. Here is again an electron micrograph of a eukaryotic cells that has a lot of structures, but again, it's not going to necessarily have everything that we show you in one of our pictures that's done by an artist. Now, do understand that the artist takes some liberties when they do these pictures. So if you look at this Golgi here and you compare it, I don't see a Golgi over here, but you compare it to, no, I don't see a Golgi there, to the Golgi from the previous structures, it's overstated. It's not quite that big. Again, the mitochondria might be a little overstated. Some of the sizes of things are overstated. This is a little bit more crowded the way we have the structures filled in than what you see over here in the electron micrograph. Again, we'll never ask you to identify something from an electron micrograph. We will give you these kind of cartoon-like structures. We want you to understand that this is just a representation and isn't a true cell, and you're never really going to find this picture by looking at an electron micrograph of a true eukaryotic cell. Going back to the things that we've looked at before, I am going to test you this time specifically on the organization that we have for living things. So you need to know the prokaryotic cells include the eubacteria and the archaebacteria. The eukaryotic cells include the plants, animals, fungi, and protozoans. Again, remember, you're an animal or you are a eukaryotic organism. Prokaryotes are the bacteria. So when we look at the prokaryotes, again, the archaea are the extreme bacteria, the eubacteria are the common bacteria. When you think about disease-causing bacteria, we're talking about the eubacteria or the regular bacteria. Again, here's some really interesting pictures of people collecting eubacteria. In this case, they're at a hot spring, and these are some eubacteria shown with a reverse image or a negative imaging photographic technique. They're difficult to see. Biochemically, they're a little bit unique. Their ribosomes actually more closely represent eukaryotic ribosomes, but they also have some very unique differences that make them different from other prokaryotic cells. Here's a gram stain, or a typical stain that we would do of a typical eubacteria, the bacteria that are more familiar to us, that you might be familiar with causing disease. These are all single-celled organisms. They have different shapes. You can see some of them are more rod-shaped, some of them are more circular shaped, some of them are spiral in shape. They live in the same habitats that humans live in. They can cause human disease, but they're also very important in terms of the breakdown of living things. When things fall to the forest floor and are recycled, it's mostly done with the help of bacteria and fungi. And so we really do rely on eubacteria for much of the work that they do in making soil, in getting rid of organic waste after something is no longer living. When we go to the eukarya, we can start talking about the protozoans or the protists. Now, the picture in the far right corner shows some algae, possibly an algae bloom that you would see in a waterway. And we look at this and it looks somewhat disgusting or unpleasant, but actually if you're able to look at protozoans under the microscope, very often you'll see that they have complex structures, and they're actually quite beautiful. And these are some of the beautiful prokaryotes, some of the beautiful protozoans that you might see under a microscope. Here's a picture of a variety of fungi. Fungi can be single-celled. Important to note that the yeast are fungi, and so The single-celled yeasts we're very reliant on for making bread. They're what cause bread to rise, or they're what make beer or wine. But they're also, fungi also make up the mushrooms. And even this lovely picture of a bread with mold on it, we tend to look at it and think of it as rather disgusting. But we actually have many antibiotics were derived from some of these molds. So we do even rely on some of these molds for useful purposes. They're actually used to make medicine. The plants are, you need to know that they are all autotrophic, and they have cell walls made of cellulose. Again, amongst some of the biggest living organisms are plants. Here's a picture of some members of this kingdom Animalia, which is part of the eukaryotes. The animals are unique because they don't have cell walls. They're always heterotrophic, and they're multicellular. Now, again, you are in the animal kingdom, but so are things like jellyfish and sponges and flatworms and worms and bugs and crabs, and then we go on to all the more complex animals. But again, we all belong to the same kingdom, but we come from different families. We are amongst the more complex of the kingdom Animalia. Let's go on and talk briefly about why cells are small. Now, you did a lab on this, so you know the smaller a cell is, the more surface area it has in relationship to volume. What I need you guys to remember, again, here's the calculations, is I need you guys to remember that things that have more surface area have more room for diffusion, so materials can move into and out of the cell by diffusion and osmosis more regularly. In just a minute we'll start talking about cell transport, but that's really important, and it helps us to understand why cells are so small. It does mean that small things have more heat loss across their greater surface area, and so do understand that surface area to volume issues are important in terms of heat retention, that large multicellular organisms can retain heat, and it helps to explain why multicellular humans can live in very extreme environments where smaller and less complex or organisms that have a smaller surface area or greater surface area in relationship to volume just can't maintain their own internal temperature. Again, just a real quick discussion of the basic organization of cells. All cells are surrounded by a plasma membrane. The plasma membrane has at least a phospholipid bilayer, but all those bilayers are going to have excuse me, proteins embedded, and they may also have some kind of barrier on the outside. Within the membrane, there's a cytoplasm. Cytoplasm is a liquidy membrane with where things dissolved in it. The cytosol is the water and dissolved molecules that make up most of the internal parts of the cell, but there are organelles, which are enclosed by membranes that carry out specialized functions. They have specialized structures. There's also large macromolecules that may fill up a cell as well, that we call insoluble particles. So let's take a moment and talk about membrane structure, and then we'll kind of call it quits for this end of the lecture and then go on to the rest in a second lecture. Again, I'm going to break this up to make sure that the computer can hold all of this. The structure of the membrane, we use something called a fluid mosaic model. So when I talk about a membrane, I'm talking about something that's fluid, not static or stuck in place. So we often talk about membranes as walls, but the membrane around your cells isn't a wall. It's something that's actually in motion. So it would be more like a tent, more fluid like a tent than like the brick walls that surround you. The phospholipid bilayer is the main component, but there's proteins floating within the bilayer, and then other molecules. Cholesterol is really important, things called lipoproteins, glycolipids, and other molecules. Again, what's holding it together, because it's a fluid arrangement, is literally interactions between the molecules in the cell. So we need to know those terms, hydrophobic and hydrophilic. We know phospholipids have hydrophobic tails, and those hydrophobic tails interacting with each other in a watery environment literally hold that cell membrane together. The neat thing about the cell membrane is that it is pretty impermeable to large and polar molecules. Take a look at this picture here This is actually something that we'll go over more in Chapter 5, but this is something that for test purposes we'll be talking about a lot and you'll need to know a lot about. So again, this picture describes this area on the outside as being the extracellular environment and the inside as being a cytoplasm, and this yellow and green structure as being sort of a wall in between them. But keep in mind that it's a fluid wall. If I were able to, with my mouse, punch into this or push down on this, that it would bend downward with my mouse. The phospholipids, okay, for the most part they have polar heads. You need to know they have polar heads. They have hydrophobic or nonpolar tails, and these hydrophobic tails stick together because the environment outside the cell is watery, the environment inside the cell is watery, and the hydrophobic molecules interact best with themselves. Okay, proteins embed themselves all the way through the phospholipid membrane, and so those proteins can serve as pores that allow molecules to move in and out. Now, the previous slide actually mistakenly said that the membrane is permeable to water. There are actually proteins called aquaporins that allow water to move into and out of cells. There are other channels that are just like this pore, but they are not open all the time. They're gated. They're only open at some times. Okay, so we call those gated channels. And there's other channels that aren't open unless energy is used to allow molecules to move in and out. They actually actively transport molecules from one side of the membrane to the other. Still more protein molecules do more complex tasks. Some of them work as part of the structure, and they bind to intracellular proteins that make up the skeleton of the protein. I'm sorry, the skeleton of the cell. There's also proteins that we call receptors. What happens is molecules outside the cell will interact with the receptors and send a message into the cell. There are recognition proteins that helps the cell recognize foreign particles. And here's a nice picture of a lipid bilayer. And finally, these little yellow structures you see in here are cholesterol molecules. Cholesterol molecules play a role in making the membrane a little bit more rigid, so it isn't just all fluid. You see more cholesterol in animal cells because we don't have an external cell wall. Okay, the cytosol or the cytoplasm contains the cytosol and then non-membranous structures. Those non-membranous structures include large particles. For example, I have many cells that have lots of fat in them. Those are large insoluble particles that are found inside some of my cells. There's also the membrane-enclosed organelles. The membrane-enclosed organelles can have a single membrane or a double membrane. I'll stop with that just to break this lecture up a little bit, and then we'll start again looking at the structures that are found within the cytoplasm.